अच्छा,
कौन सा वाला सर बताइए?
सिस्टम्स बायोलॉजी पार्ट 2 भी इसको खोल दीजिए।
और ये
हाँ, ये इसको भी खोल दीजिए।
पहले पार्ट 2B को पढ़ाएंगे या पार्ट 2?
पार्ट टू कल कंप्लीट नहीं हुआ था, उसको कंप्लीट करने के बाद फिर 2 में चले आएंगे।
2B में जाएंगे। ओके।
ये कितना डिले चलता है?
5 to 10 सेकेंड।
ठीक।
अभी आप पार्ट 2 का इसी से पढ़ा रहे हैं।
हाँ, तो उसको भी एक बार खोल देखिए, तो खुला हुआ है क्या?
ये पार्ट 2 है।
हाँ, वो भी खुला हुआ है।
ये पार्ट
नहीं, ये भी सेम। अच्छा, दिखाइए फिर वो।
ये पार्ट 2 है। आप इसको मिनिमाइज करके फिर आप पार्ट 2B को खोल देंगे उस टाइम।
ठीक है, ठीक है।
मैं इसको खोल देता हूँ फिर भी। ये है।
हाँ, ठीक है।
ये मिनिमाइज करके
हाँ।
आपको इसको क्लोज कर देता हूँ। अभी आप पार्ट इसी को
हाँ, इसमें
एक एक फाइल करिए, वो
ठीक है।
बता रहे हैं। क्यों आ तो गया स्टूडियो 3 का ना? हाँ, तो क्या दिक्कत है? आया नहीं? आ गया तुम देखो ठीक से। सर
सर,
अभी आप देखो, आ गया ऑडियो आ गया।
hello, good morning. So we are going to start in next 2 minutes, okay? ठीक है।
स्टार्ट कर।
हाँ, मैं देख ले रहा हूँ, जो हम लोग का पिक्चर भी आ रहा है।
हाँ आ रहा है ना आपका
क्या भी?
ये ये वाला छोटा ये आ रहा है ना?
अच्छा, ये वाला है इसको। अच्छा
आप जब पीपीटी करेंगे वो बड़ा जाएगा।
अच्छा, वो
आप पीपीटी पे चलिए।
ठीक है। अच्छा, कितना जन लॉगिन हुआ है ये कैसे?
वो लाइव चार्ट में आएगा क्लास में।
अच्छा,
आपको दिखा दूंगा सर, अभी आप क्लास शुरू करें।
ठीक है। यहाँ पे लाइव स्ट्रीम ऑफलाइन दिखा रहा है।
ये सब देखो ना, स्टूडियो आप एक बार रिफ्रेश कर दीजिए। ये देखो ना सर।
अच्छा, ठीक है।
अभी आपका नेटवर्क थोड़ा इशू है।
इशू है।
डाटा ऑफ है, ऑन है ना?
डेटा तो ऑन है।
आ रहा है सर अभी।
ठीक है, मैं स्टार्ट कर देता हूँ। 2 मिनट में मैं स्टार्ट कर रहा हूँ। Hello, so we are going to start right now, okay? Okay, very quickly. So today's class I'm going to go a little fast because yesterday it was a little hard for me to you know start with this one because this is a completely new concept for us. So today we are actually going a little fast, assuming that you people don't have too much trouble in understanding yesterday whatever we discussed about. So starting from that, so we are going to start right now, okay? Okay, very quickly. So today's class I'm going to go a little fast because yesterday it was a little hard for start with this one because this is a completely new concept for us. So today we are actually going a little fast, assuming that you people don't have too much trouble in understanding yesterday whatever we discussed about. So starting from that, so we are going to start right now. Okay, so yesterday what I talked about is linear systems and non-linear system. So I had also explained to you that biological systems are non-linear, and when we say non-linear, this means it is very hard for us to predict the outcome of the system just with a few initial values. Now, if it is a linear system, it would have been much easier, but being a non-linear system, it is relatively hard for us to conclude what is going to be the final outcome of the system. And I had given you an example of yesterday. I had given you an example that if you have two different cytokines, and I actually explained to you also about an experience when I was actually doing research that someone was actually doing a treatment with two different cytokines, and then she was also supposed to do a co-treatment where they're going to add two cytokines together, but she forgot to do that one, and then she took the two individual data and then tried to digitally or mathematically add these two together, but eventually it doesn't work because if it is a non-linear system you cannot add 1 plus 1 and get 2. So that is the caveat of this non-linear complex systems like biological system. Just be aware of that, okay? Now we are going to proceed from here. This the thing that I was actually trying to tell you, that this is experimental setup where we have to do either separately or actually together. So I'm just going to rush with that slide a little fast and bring to this slide. Now in this slide, if you see, we have a image where I actually showed you two different kind of graph. One is them which is a linear one where it is a highly predictable one where the input and the outputs can actually be matched with each other. So in other words, if you have an input and if you gradually increase the input, then you also see a gradual increase in the output. Now the biological systems are actually non-linear, so they follow this kind of a nature, a rather sigmoidal nature, not a linear nature. This is again because of this non-linearity in the systems, and here you could see that the output and the input really they don't match with each other. So in other words, you may not have an equivalent amount of output for an amount of an input. So this is something what is actually written over here, that the non-linear systems has unpredictability and complexities and a lot of entropy. We didn't have to understand about entropy, forget about that right now. Just remember these two terms, that non-linear systems has a lot of unpredictability and complexity. Okay, and the non-linear systems is hard to deconvolute. When you say deconvolute, this means if you have a linear system, it has a capacity to predict what is going to happen in the next phase, because this is a fairly straight line. But in the case of a non-linear graph, you could see it's a sigmoidal S-shaped curve, and over here it is literally hard for us to predict the outcome simply by knowing the input of that one, because it doesn't follow a linear kinetics. Okay, that much we need to remember, because this is kind of a preferred question in the exam, that what is a linear system, what is a non-linear systems, biological systems are linear or non-linear, explain with a graph or some kind of a representations where we know that it's a non-linear system, not a linear system. Okay, so I'm going to leave that one from there and proceed to the next one. Okay, this is a sequence of a genome. Now we are actually a little jumping from what we have been talking about, because I'm going to give you a kind of a fairly bird's eye view of this system's biology rather than going incrementally every step, because that way we need a lot of classes and also a kind of an actual classroom, not a virtual one, to explain to you on a blackboard. So I'm just going to pick up a few examples and try to give you an idea that what is exactly the non-linear systems and how complex the biological systems is and how systems biology is going to be helpful for us to understand it. Okay. Now this is a genome sequence. Now all of us know that whatever the living organism you take it, doesn't matter whether it is a fish, whether it's a bird, whether it's a human being, whether it's a bacteria, virus, it is going to be after all composed of the strings of 4 different nucleotides, right? That's the maximum you have, ATGC. It's their combination which actually produces the different genome sequence for the different organisms, right? Okay. Now why I had put over this one to just tell you that if I give a genome sequence like this and ask you a very simple basic question, that can you predict the organism which is encoded by this genome, or in other words, this is the genome sequence of a human being or a fish or a plant or a bacteria or a virus, what is it? So just by reading the genome sequence, why I pose this question, because just by reading the genome sequence, it will be very hard for us to correlate with an organism. Or in other words, you take any organism, sequence it, you will have this kind of a jumble of millions and millions of ATGC, strings of ATGC. That really doesn't tell you that what organism is actually been hidden in that. Okay, I'm going to show you the next slide and then again come back to this one. Okay, so the next slide is that pufferfish. So this is the genome sequence of a fish that is known as pufferfish. Now, why I had again showed you, because of the fact that unless and until we add something extra to the genome sequence, it is not possible for us to directly reach to the conclusion that what organism's genome is this. Okay, or in other words, you can have this kind of an strings of millions and millions of ATGC, but within this, any animal can be hidden. It could be a genome of a monkey, it could be a genome of a plant, bacteria, human beings, doesn't matter. So just the genome sequence cannot tell us, or we cannot go back and predict what organism could possibly erupt from this genome sequence. Or in other words, again, if you take the genome sequence of, if you take a fish and do the sequence of it, you might get this one. So from the fish you can get this sequence very easily, but from this sequence it is impossible for us to tell that this actually encodes for a fish only and not any other millions of species which we know that exist in the earth. So it needs not only just the genome sequence, but something which is a little more than this information. Okay, so that is something where the systems biology is helpful, because it is just not a linear correlation between these two. So this is where I'm going to bring to a different slide, and this slide actually also will tell you the variabilities which actually happen when you do an experiment. So this is just an example of an experiment which we kind of carry out in our labs and see the amount of variability which we could. So far whatever the slides I actually showed to you, I just wanted to give you this feeling that the biological systems are a little hard to predict. Sometimes it is very hard to predict because of the reason that we don't have a linear relationship between the events that is happening in the biological systems. So these are not a linear relationship, these are non-linear relationships, and in that case it is sometimes very hard to predict the outcome of the system just having a knowledge of a few of the inputs. Okay, now if you see over here, this is an example of what I actually was trying to tell you. This is another example. Okay, now this is the way in which most of you are going to possibly start growing the cells in your labs whenever you are going to do the research. So you go to grow the cells. Say for instance, in this case I took an example of HeLa cells, right? This is an HeLa cells. This is a cervical cancer cell line from one female patient who was having cervical cancer. This is the cell which has been most extensively studied in biology, these HeLa cells. Okay, I just gave that example. The cell has not much to do in giving the example. Just follow the different steps which I am trying to raise over here. So you take the cells from the cervical cancer patients, and we have this cell line available with us. And then what you do is that you take that cell and then put it into a petri dish. You know, this is kind of a culture media, culture plate where you kind of grow the cells. And if you go back and see the manufacturer of that plate, possibly someone sitting in Vietnam has actually manufactured the plastic wares, or maybe it is actually been made by Chinese companies in China. And then you take these cells and then you grow them in, whenever you grow the cells, you add the fetal bovine serum. This is the source of proteins and the cytokines for the cells to grow. So any cell you grow, you need to grow them in the presence of FBS, which is fetal bovine serum. Now if you try to grow them in the fetal bovine serum and then look into the bottle where from it originated, possibly it has been originated from most of the fetal bovine serum which we receive from India, which we receive in India, it actually comes either from Australia or New Zealand. So the tissue culture plate where you're growing the cells it has come from Vietnam. The fetal bovine serum which you are growing possibly has come from Australia or New Zealand. And that whenever we grow the cells, we add antibiotics so that the bacterial infection doesn't come over here. And that particular antibiotics possibly is made in Pune, And the media where you grow the cells has been made in factories in Gurgaon in Haryana. And then we are keeping the cells after you add everything. So you are keeping the cells into an incubator, and possibly these incubators are made from thermo companies, and these are coming, imported from United States or something like that. So what I'm trying to tell you is that think about all these conditions, that one petri dish plate, it came from Vietnam, the serum came from Australia, the media came from Haryana, Gurgaon, the antibiotics came from Pune. So all what you're doing is that you are actually adding a cocktail of different things that has actually originated from different parts of this world. Now suppose your friend is trying to do the exactly same experiments, but he or she has been actually using the ingredients which has originated from a different place. Suppose they are getting the fetal bovine serum not from New Zealand or Australia, but from South America, and they are getting the antibiotics maybe from Mexico, not from Pune. So in that case, we are introducing so much of variabilities right at the beginning of the experiment. See, we didn't even start the experiments. The experiment is yet to start, right? This is where the experiment is going to start. But before the experiment even start, the very act of preparing for the experiments, growing the cells and then culturing them, maintaining the cells in a tissue culture media, there are so many variabilities over here because each of us actually use a different lot of chemicals, different ingredients purchased from different vendors, and therefore all of these variable factors has an influence on these experiments, and therefore it is very hard to reproduce the experiment what you are doing with your friend or colleague has been doing because they had a different sets of conditions. This is again trying to enforce on you whatever I was talking so far, that is the non-linearity of the biological systems. The biological systems inherently are so non-linear that it is very hard for us to predict the outcome of it just by knowing what is going to be the input, right? And on the top of it, when we do the experiments, we further introduce lots of different ambiguities within them, lot of different confusions and lot of different uncertainties into the experiments. So that is actually adding up to the already existing uncertainties because of the non-linearity of the systems, and together this becomes very hard, and therefore it becomes very hard for us to do the reproducibility. And you yourself can test it. I mean, you do an experiment today, you do an experiment tomorrow, you don't have to wait for next month or next week, just do it tomorrow. You will see that there will be a subtle change in the readings. You will not be able to reproduce exactly the same reading whatever you have generated today. And all of this is because of the fact that there are so much of variabilities in the system, inbuilt system, because of the non-linear nature. And in addition to that, when we do experiments, we introduce a lot of variabilities. So this is again where systems biology is going to come to the rescue, because what we are going to do in systems biology is that we are going to collect large number of data sets and then look into the statistical significance of those data sets taking into consideration of these variabilities and then give us an interpretations. But if you're not doing systems biology and if you're just trying to follow it up linearly, then you might land up with an issues of reproducibility and getting this and not getting the same data back tomorrow when you're going to repeat the experiments. So be aware of that. I mean, this is something just to tell you, to make you aware that these are the problems which most of the researchers actually runs into whenever they do an experiment without realizing that what kind of variabilities they had actually introduced within the system while doing the experiments. Okay, so that is again one of the fallouts of the non-linearity of the systems, of the complex systems. Okay, now this is something what I was trying to talk about you in a few minutes back when I showed you the genome sequence and asked you that okay, this genome sequence predicts which organisms, or if you have a genome sequence, can you tell that whether a tiger is been hidden within the genome sequence, or a bird is been hidden in the genome sequence, or an apple or a banana is been hidden in the genome sequence? What does this sequence? Just if you have the sequence, can you predict the organism, or can you go back and tell us that this organism actually came from the genome sequence, not the other way around? Okay, it is very hard for us to tell. That's what I was telling you, because so many different organisms can be possibly predicted from that particular genome sequence that you will be completely confused, because you may not be able to tell precisely that this organism's genome sequence is this just by looking into the sequence of this ATGC. Now this is something what we call as an emergent property. Again, very important term to learn into systems biology. Emergent property is the property where a new phenotype or a new property actually we get it not because of the reason that this has been directly evident from the sequence or from the informations, but it is because of these interactions of the systems with the surrounding. Remember, to yesterday's class we were talking about so many different things. We are talking about systems, we are talking about ingredients, we are talking about constituents and components, we talked about something called the interface, boundaries, strain, all of these terms. So this is again something which is related to that emergent property arises because of a complex interactions between the system and the surroundings. And as a result of that, when you see a phenotype, it really doesn't correlate directly from the genotype. It has to do more with the genotype. And this is where we talk about what we call as the networks and other kind of complex things, because I'm going to give you an example and show you that what are networks, how a gene actually operates within a network, not in isolations, not in vacuum, and how this emergent property actually really emerges, right? But just for this time being, for now, so far with these slides, you just remember the very important term, that is emergent property, which is a new property which is not directly predictable, quote unquote, from the data, but because of the reason that this system actually interacts with the surrounding, so we get a different kind of a property that is known as the emergent properties. Okay, don't forget that. Now I'm going to skip this slide. The next slide I'm going to skip it because this is not very relevant for us. So I'm just going to, but having said that, if you are interested in knowing about the slide, feel free to go through the slides, because either way I'm going to pass on you the slides. But right now, for our exam point of view, this is not important. So I'm just skipping this one. Okay, I'm going to bring to the third example of this one. Let the slide to copy. Okay, this is the biopsy or whatever it is of the brain of a section of the brain. Okay, this is the biopsy or an histological sections, whatever you call of a part of the brain or a slice of the brain from an individual who is suffering from Huntington's disease. All of us know that Huntington's disease is a neurodegenerative disorder that affects the muscle coordination and leads to mental decline and behavioral symptoms. Okay, so we are not here to talk about Huntington's disease. I'm going to talk something a little different from that. So feel free to go and read about Huntington's disease if you are interested about it. So that is not the theme of the class. The theme of the class is, over here we have two slices of the brain. One is been obtained from an individual which is absolutely healthy, normal, and another is from an individual who is been suffering from Huntington's disease. Now if you look into them, we don't need we don't need a pathologist, or you don't need an anatomist, or anyone, to tell that which brain actually belongs to which individual. So obviously this one which I'm showing my pointer right now, this is the brain section of the brain from an individual who is perfectly healthy, and this is the section of the brain from an individual, slice of the brain from an individual who has been suffering from Huntington's disease, right? So if you see over there how much dramatically they're different from each other. This is another section which will show you that this is the brain of a healthy normal individual, and this is a slice of the brain from an individual who is suffering from Huntington's disease. Just look into this versus the control. You will appreciate that how much the brain has been shrunk by the size, how much it has been smaller in dimensions, and how much it looks very dry and completely been kind of a necrotic kind of a view if you compare it with the control. So we don't have to go to a pathologist to know that one. I mean, just looking into the sections itself, you will see from the morphology, from the phenotype, that the brain from a Huntington's disease has undergone an atrophy kind of a thing, right? It's been shrunk into size and shape and everything. But if you go back to the biology of this disease, then there is just one gene which has been affected. That is known as Huntington's gene, or Huntington's gene, HTT, which actually codes for this protein, HTT protein. Now what happens is that there is a trinucleotide expansion, and then as a result of this trinucleotide expansion is one of the trinucleotide expansion disease, and because of this, that's the root cause of the pathology, that the size actually goes on increasing, and the length of the gene actually keeps on increasing because there is an expansion of the CAG repeats, and it becomes so large that the RNA polymerase cannot transcribe it, and eventually it leads to the loss of this particular gene, that is HD gene. Why? Because of this reason that there is a trinucleotide expansion of the introns of this gene, and as a result of this, the RNA polymerase cannot efficiently transcribe this gene, because now these introns are super large in size because of this expansion, and therefore the RNA polymerase fails to transcribe the gene, and this gene is not been encoded. But the question actually starts from there. Why does, that's what is actually written over here, why does so again, this disease is very lethal, still we don't have any cure. The question is that why does the disruption or the non-functionality of just one gene will have such a profound influence on the phenotype of this entire structure? Or in other words, why should just one mutation of a gene should affect the phenotype so dramatically? What is the reason for that? Well, again, the reason for that, if you scratch the head, you will not be able to correlate or find out the reasons directly, but if you use the concepts of systems biology again, it will help us to draw some conclusions. The reason for this is possibly right from the phenotype. You could possibly make the conclusion that this particular gene, that is Huntington's gene encoding this HTT protein, is important as a hub for different cellular signaling pathways. Or in other words, if you don't have this particular gene, then multiple signaling pathways actually collapses, they don't operate, and therefore this gene appears to be a master regulator controlling so many different genetic circuits. Just because of this reason that this particular master gene has been lost, or has been absent, or has been mutated, is not functional, so the entire network actually collapses. Network means all the surrounding things, right? It is something like this, that if we remove, or if we close the director's office at AIIMS, I'm just giving an example. If you close or remove the director's office at AIIMS, what is going to be the repercussions of that? What is going to be the consequences of that? Well, we know that when that office closes, then most of the things related to that office, dependent on that office, is going to close, right? The OPD is going to close, the library is possibly going to close, the research section is going to close, the different departments are going to close, because that's the main nerve center that controls and runs the entire hospital. And if that closes or that shuts down, it is going to have its effect globally on almost all the departments at AIIMS. Exactly the same way, this Huntington's gene has been so vital because it actually sits as a hub of a signaling cascade, and it has been acting as what you call as a master gene. And because of this nature that it is a master gene, it actually regulates multiple signaling pathways. And therefore, if this is gone, then all of those signaling pathways, all of these networks, all of these different transcriptional activities surrounding this, which is dependent on this particular gene, collapses. They no longer exist, and therefore you will have a profound influence of this in the entire brain. And that's exactly what you're seeing over here, that just removal of one gene has such a large dramatic consequences on the entire structure, because of the reason that this has a huge fallout, just because of this reason that that particular gene is the center of a very important cascade of the signaling pathways. So this is again part of what we call as the gene network. Remember yesterday we were talking about interconnected, so this is something like Sajib, is it still stuck in the same slide?
স্যার একবার করে দেখছি দাঁড়া একটু হোল্ড করুন
Yeah, sure.
এখন স্যার মানে বন্ধ করেছেন কি কাজটা
Hold on, let me see.
আপনার কোনো স্লাইড তো দেখাচ্ছে না এখন এমনি ওই যেটা ব্যাকগ্রাউন্ড মানে শুরুতে যেরকম দেখায় সেরকম দেখাচ্ছে
Now
এখনো কিছু দেখাচ্ছে না
There's-
স্টপ স্ট্রিমিং করতে বা যেটা করতে
there is a delay of a couple of seconds.
স্লাইড আসেনি কিন্তু এখন
Acha, I had already put the slides. See? Still same?
এখন আপনার স্লাইডে এসেছে নেটওয়ার্ক কোলাপস এই স্লাইডে খোলা আছে কি
Now
চেঞ্জ করুন স্লাইড একবার চেঞ্জ করে দেখুন স্যার
now it changed it. See?
চেঞ্জ চেঞ্জ কিন্তু হয়নি স্যার এখনো
5 minutes 10 seconds delay. Just see.
সেটাই মানে একটু ওয়েট করছি not yet
This is a big problem. Actually, MDs are around, right? MSCs are not coming. That's the only thing.
হ্যাঁ না এমএসসিও আছে স্যার
No. They are not coming physically, right?
ও না এটা মানে অনলাইনে ক্লাস করছি
I don't know which is the best system to do. Not yet?
আচ্ছা এখন হচ্ছে চেঞ্জ সাম ফেনোমিক্স জিনোমিক্স একটা স্লাইড আছে
Extended phenotype?
এক্সটেন্ডেড ফেনোটাইপ হ্যাঁ স্যার চলে এসেছে ঠিক আছে
Okay. But the thing is that there is a lot of delay.
ঠিক আছে স্যার কন্টিনিউ করুন তাহলে আপনি আছি
Okay. Hello. हाँ गुड मॉर्निंग अरे पता नहीं आप कार्तिक से बात कर ले एक बार Hold on, so let me get an feedback from you people because I'm not aware of what's happening. It should not happen that I completely complete the class and then nothing is. Should not happen that I complete the class and then nothing is. Okay, so in between, I just received seeing this feedback from you people. Extremely sorry because what happened is that the network froze, in between, and as a result of this, a couple of the slides which I'm supposed to show you actually got lost. So is the audio also lost? Did you hear it about it, or is that you didn't even hear it also? So because even if you hear it, then I'm not going to show the slides again. I'm going to. I suggest seeing this feedback from you. Extremely sorry. What happened is that the network froze, in between, and as a result of this, a couple of the slides which I'm supposed to show you actually got lost. So is the audio also lost? Did you hear it about it, or is that you didn't even hear it also? So because even if you hear it, then I'm not going to show the slides again. I'm going Okay, so it seems that the audio was also lost. So, okay. Okay, so this is the slide which I'm going to fix it, and I'm going to. So, the moment if you can't listen or hear or see the slides, just give me. Yeah, Kuntal, is it audible now, Kuntal? I'm speaking pretty loud. Is it audible now? So this is the slide which I'm going to fix it, and I'm going to. So the moment you can't listen or hear or see the slides, just give me. Yeah, Kuntal, is it audible now, Kuntal? I'm speaking pretty loud. Is it audible now? Okay, so what I was talking a little, few minutes back. I'm just going to extend the class for another 10-20 minutes. Bear with me because I never anticipated that is going to be happening this problem. So just bear with me couple of slides so that at least I can finish up the today's class. So what I was talking about a little back is that this is something called the extended phenotype. What is that extended phenotype? Well, that is something which is that if you just have an organism and just look into the genome sequence of the organism and try to draw all the conclusions from that genome sequence, then we might not be able to draw all the conclusions from it because of the reason that this particular genome sequence of an organism, which is regarded as a blueprint of an animal or blueprint of an organism, interacts in a complex way with the surrounding. And therefore, finally, when you see the phenotype or finally when you see the behavior, then that is different than that what has been encoded just within the genome sequence. So why I had put this slide to show you that this is a slide of a bird which has built its nest, right? Think about it as any bird. It's a cuckoo or eagle or kite or crow or whatever it is. We don't care about that. But what is very important is that if you take, suppose this is the nest of a cuckoo bird, and if you take a cuckoo genome and then look into it, that is there any region of the genome that actually tells it to build the nest, you would see no. There is no region of the genome, there is no protein, there is no gene which actually encodes and tells the cuckoo bird to build a nest. for that matter, even if you take human beings, like if you take the human genome sequence is actually been sequenced in length and breadth, right? And then if we kind of look into the human genome sequence and try to draw the conclusions about all the human behaviors, then we may not be able to do it ever. Like we build house, right? We drive cars, we like to have families, kids and everything. But if you see, look into the human genome, we really don't have any portion of the genome that actually encodes us. We don't have any gene, any dedicated protein that tells us to build a house, to drive a car, to have a family, kids, you know. So all of these are actually something what we call as an extended phenotype which is not been written into the genome, but that actually emerges because of something called the emergent property. And why it emerges? Because this genotype actually interacts with the surrounding, and as a result of this we have this complex phenotype. And this is what we call as behavioromics, which is study of the behavior of an organism, just not phenotype, but it is a behavioromics where you understand the behavior of an organisms when the genotype or the genetic makeup of this organism interacts with the surrounding in a non-linear fashion. And this is again part of the systems biology, where we try to understand not just what is written within the genome, or what not just what is actually evident from the experiments, but also what is actually been being but what emerges out because of its interaction with the surrounding. Okay, so I'm not showing you the next slide, which is the summary of the today's class. So again, we missed out this slide. I'm going to again, go through this one because this is very important. This is kind of concluding whatever we actually studied in the part 1 of the systems biology. So again, give me a feedback by writing a message in the chat box that whether you are going to listen or able to listen me or not. So are you going to able to hear me? Are the slides frozen, or you can still see them? So just give me a small feedback in the chat log, and then I can proceed with the summary of this class. Okay, so I'm going to draw to the last slide, which is kind of the summary of today's class. So the first point, so over here I had actually mentioned a couple of things. There's something which you must remember and something which is okay to remember, not very important, but you could still remember it. But something which is must remember is you should remember definitely, because that's something which is the core philosophy of this class. Okay, so systems biology is a way to analyze the data sets. So again, I'm telling you, and again kind of hammering you in your brain that again and again, when you try to say the systems biology, don't think that we have to open a new building, we need to recruit new faculties, and we need to have a new establishment completely to teach you systems biology or to take you through the systems biology. It is a way in which all of us can actually start practicing when you understand that these are the complex things which could happen in the background, which most of us kind of ignore it. Remember the example which I had told you a few minutes back about how someone is growing a cells in a tissue culture flask, and what are the different variabilities that actually can come in the experiments and can completely modify the outcome of the experiments. So if you are doing it in a particular way, and if a friend or colleague is doing it in a different way, and if your variabilities are different, then you are going to end up with different results. So systems biology, more than a new subject or a new domain, it is not just a subject but also a philosophy to rethink and alter the mindset. So how you are going to look into a data, what are the precautions you are going to take, what are the things which you are going to factorize when you are going to set up an experiments, how you are going to design the experiments, whether you are going to think that these are the factors which could modify the outcome of my experiments. So all of this actually comes under the domain of the systems biology. So it's more of a philosophy, it's more of an approach, it's more of a mindset for you to attack the problem, yeah, right? Rather than starting something brand new. So that is something which is very clear, and that is something which you must remember. Now, the next point is that systems biology relies in a vast number of interactions between the components of the systems. Remember, this is the example which I had actually given you with the Huntington's disease. So, in the Huntington's disease, you will see that there is just the mutation of one single gene, the Huntington's gene, and then that particular has a profound influence on so many gene networks which has been related to that. And therefore, if that particular gene has been lost, all of those networks which has been related to this particular gene collapses or it gets lost. It is something which I had given an example. It's something like the director's office or the director's building. If you stop the director's office, or if you close the director's building, then what is going to happen is that it's going to have its effect on the OPD. It's going to have its effect on the library. It is going to have its effect on the different departments, because that's the nerve center, that's the central point what controls the entire institute. And if we just remove one office, it may be just that we are removing only one office, but just if you remove one office, then it would have a profound influence on the entire institute and its function and how it functions. So that is what is actually very important, that there is a lot of complex interactions between the different components of the systems. Remember, when I was talking in the yesterday's class, we were talking about interconnected, interrelated, interdependent, interacting components, right? We had mentioned about these words: interrelated, interdependent, interconnected, and interacted components, right? So these are all the parts of the systems biology where there are a large number of interactions that takes place between the components of the systems. And if we ignore that, we are not going to get a good idea about what is the final outcome of the systems. So we must remember that. And the example which I have given you is this with the case of a Huntington's disease. That a single gene mutation has a profound influence. Now, 10 years back, people never used to figure out that why it is something like that. This is completely unprecedented, that one single gene, if it has been gone, why should it have such a profound influence on the functioning of almost entire brain? Now we understand that, because that's the master regulator gene, and it actually controls so many different gene networks. So if that is gone, that is lost, that is deleted, if that is defective, or if that is mutated, then it is going to affect each and everything which is dependent on that. So it is a question of interactions and interdependency between the different components of the systems. Okay. The third point is that systems biology is important since biological systems are non-linear. Again, we had given you the example that what is linearity and what is non-linearity. Non-linearity means where you cannot predict the outcome just by looking into the inputs of it. That is what is non-linearity. And linearity is that where you could do a regression line and you could actually always predict what is going to happen to the future if you know that this is going to happen. This is what is actually happening in the present tense, right? It is something like prediction of the weather. You cannot predict what the weather is going to be there. You cannot predict with 100% certainty. You could predict still, but not with the 100% certainty what's going to be the weather tomorrow, or say 1 month after now. The reason being is that because there are so many different factors that actually regulates the weather at a particular place, that it is just impossible for us to draw the conclusion of it entirely just by reading a few data today, right? So this is an example of a non-linear systems where the output and the input does not correlate in a linear fashion. And to be very important is that majority of the biological systems are actually on non-linear systems. So that is exactly what has been mentioned in the point number 4. The non-linear systems are different to predict and quickly descend into chaos. So if you have non-linear systems, you cannot predict them very efficiently, because there is a complex interaction that takes place not only between the components of the system, but also between the systems and the surroundings. We are talking about boundaries and interfaces and so forth, right? And therefore, because so non-linear systems are difficult to predict because of the reason that there is a complex interaction between the components which makes up the system and the surrounding or the environment, and therefore it is a little hard for us to predict. And then we had also talked about emergent properties, extended phenotypes, something which pops up from the genome sequence, but still it's not been there in the genome sequence. So per se, this is something what we call as the behaviomics, where the behavior of an individual is not being just encoded into the genes, into his or her genes, but rather it is actually being an output of its complex interactions with the surrounding. Okay, and there are so many examples to tell that one. Like for instance, one example which could, just I always cite into this in this class is that, if you see the human evolutions, and before the evolution actually, long before, when humans actually started living together as a society or as a colony or particular clan, then we have these individuals who are actually been as a nomadic, small nomadic tribes, and they used to go to the forest and used to hunt animals, right, for the food. Now what happened is that when they used to go outside and hunt food and hunt animals and bring it back and eat, then they always used to run into uncertainties, because there are few days when they would have ample amount of food, and they will have weeks and months when they will have absolutely no food, and they go out and they literally go for starvations. So they thought that, what, this is a very an chaotic way of leading the life. So why don't we do one thing? Why don't we stop all these kind of things and don't take it as a priority, rather start into agricultural things? Because if you do agriculture, if you start growing crops and plants, then you have a very predictable way of getting food, because every day you know for sure that this plant is growing right in front of my eyes, right under my control. By hunting the animals, you don't really know whether you're going to get an animal today or not. Therefore they started growing the cereals and other kind of things, and that is how people started cultivation and eventually started into agricultural and farming. And when they started doing that, then they say that, what, why don't you also start domesticating a few animals, like cows and buffaloes and goats, because these animals are a good source of proteins like milk, and we can drink the milk and get nourishment out of it, and it is a more predictable way of getting energy, right, rather than again into uncertainties of going and hunting for the animals and never know when you are going to get the next animal. So they started kind of domesticating these cows and buffaloes and sheep and other kind of an animals that give them the different products like the milk and the wool and all these kind of things. Now when people looked into the genome sequence and all the proteins which has been encoded by these people before domestications, after domestications, then they found that before domestications they had all the enzymes expressed at a very high level, which has been important for causing the metabolism of meats and meat proteins and those non-veg proteins. Now at that time they had a very little expression of these enzymes, which is important for metabolizing the lactose in the milk, and therefore those individuals used to have a lactose intolerance. But after they made a transition in the lifestyle and then actually converted from this nomadic life to a more systematic society life, where they had growing where they are following this agricultural practice and domesticating the animals, and then they started kind of giving a less priority to the hunting and more priority in rearing those animals, and started drinking the milk and everything, then slowly you find that the expression of this enzyme, which is the important for lactose metabolism, those enzyme actually starts getting expressed later on. So you could see very nicely people has elegantly showed that there is a dramatic change in the switch over of the enzyme activity. That is, the milk metabolizing enzymes started at a very low, and eventually the moment they started picking up, that is also the time when people also started domesticating the different animals, because milk is now the source of the food which they could actually easily get it. And before that, milk was not a source of the regular food, so they had a lactose intolerance and other kind of metabolic problems. So this is how again you could see that how complex these interactions of an organism with the surrounding it, and how that could actually give the feedback and could profoundly influence not only our behaviors but also the genetic circuits. So all these feedbacks which we get from the surrounding goes back into the genome and actually modifies the gene, and that is the reason why we talk about something called the fixation of a particular phenotype. So if an organism, you take it and then actually take it to an extremely high altitude, then you will see that the phenotype you will get fixed. What is the phenotype? The adaptation to an extremely low oxygen tension. And in those animals you will see that these pathways which are actually helps us to adapt in hypoxic conditions, like the HIF pathways, hypoxia inducible factor, those pathways get super amplified. The reason being is that now they are actually been acclimatized to staying in a low oxygen tensions, and therefore the entire system actually changes, and it has actually adapted or getting adapted to manage with low oxygen pressure or low oxygen tensions. So if we take the genome sequence of an individual staying at an extremely high altitude compared to that of the individual staying in the plains, you will see that there is a dramatic difference in them. The reason is that they had actually adapted to a different environment, and those environmental signal has been transmitted back to the gene, and the modifications of the genetic circuitry actually taken place accordingly. So that is something what we call as the complex system of interactions between the systems and the surrounding, and this gives rise to the emergent properties, or what we call as the phenotypes or the behavioral where there is a change in the behavior, difference in the behavior because of the reason that how they're actually been interacting with the surrounding. So I thought of taking the next part of the next class, but already we are actually so late. I'm going to stop over here. Otherwise, it will be a little saturating for you, and then things will not be very clear. So what I'm going to do is that I'm going to continue it in tomorrow's class, and is there anything which possibly needs to be rediscussed, and kind of we can go over again, let me know. Why don't you kind of put your queries over here, and then I know for sure that these are the things which we have to redo and repeat it, and then you can start with that one tomorrow right from the beginning.